Now, my very dear students, today we are going to talk about receptors. You know, receptors are very special macromolecules which are present within our body, and different ligands work on them. Now, we have to first discuss what is ligand, what is receptor, right? Ligands mean the substances which bind with receptors, the specific receptors. For example, all the hormones in your body are acting as ligands. Hormones. All your neurotransmitters are also ligands. Then all the drugs you use, they also bind with certain receptors, so they also work as ligands. So we can say all your hormones and neurotransmitters and drugs and even toxins, all of them bind with specific receptors to produce their effects. Because they have to bind with special receptors, right? So we call them ligands, toxins, and even some other chemical molecules, chemical substances. Now all these ligands have to work on a receptor. Let's suppose this is a receptor, right? Now what really happens, that receptor has one point to which a ligand binds. These ligands are supposed to bind with a very unique point, and this point where these ligands bind, these points are called ligand binding domains. What are they called? This is a receptor, and this part of the receptor to which a ligand bind, right? This part of the receptor is called, yes, ligand binding domain, right? Now, once a ligand bind with ligand binding domain of a receptor, then receptor is supposed to produce some biological actions. Receptor will produce certain biological actions, biological activity, biological actions, or biological activity in the body is modified. Now, again, let me repeat. What is receptor? Receptor is a macromolecule. Another way to define receptor is a macromolecule to which a particular ligand binds, and then receptor undergoes some transformational change and produces biological effects on the body. The point which you have to remember is that if hormones or neurotransmitters or drugs or toxins or chemical substances bind with some other substance, right? But this does not result in any change in the body or biological function, this should not be called receptor. Look, for example, there are plasma proteins. Many drugs or hormones or toxins or neurotransmitter, they tend to bind with the plasma proteins. But once the drugs or chemical substances or hormones, when they bind with the plasma proteins do bind with these ligands, but plasma proteins don't produce any effect on the body. Because plasma proteins do not produce any effect on the body, so we say plasma proteins are not receptors. Is that right? Again, let's repeat what we have discussed up to now. Let's recap. What is a receptor? Receptor is a macromolecule. Receptor is a macromolecule in a biological system to which a certain ligand binds. Ligand may be a hormone or neurotransmitter or drugs or toxins or other chemical substances. And once the ligands bind with a specific receptor, that will result in some biological modifications in the organism. Is that right? Now listen, if you don't have a clear concept of receptor, you really don't understand how the hormones work in your body. You don't understand how neurotransmitters work. You don't understand how the most of the drugs work. Even you don't understand how the most of the toxins work. So if really you want to understand actions of hormones, neurotransmitters, drugs and toxins, fortunately or unfortunately, you have to understand how the receptors work. What are the receptors? What are their classes? How do they work? Right? Now, First of all, we'll talk about two basic types of receptors. You see, all the substances which are going to work on the receptors, we have given a name of ligand to them. Now, all the substances which have to produce an action on a cell, they can be broadly classified into two groups. For example, let's suppose here is a cell. Yeah, this is a cell, right? Now on this cell, let's suppose there's a hormone number 1, hormone number A, and here's the hormone number B, hormone A and hormone B. Now these two hormones have to work on this receptor and modify the action of this cell. Again, there's a hormone A, there's a hormone B, or ligand A or ligand B, and these ligands are going to act on this cell and modify the function of this cell. The question is that there are two types of basically ligands, some which are large molecular weight, and some of them are highly, yes, polar, highly charged. Now, those ligands which are large molecular weight, like peptides, for example, peptides, or those ligands which are highly polar, like catecholamines, they are highly charged molecules. They cannot dissolve into lipid membranes. Because these substances cannot dissolve in the lipid membrane, so both of these substances, they cannot enter into cell due to their big size or their high charge. Because they cannot enter into cell, so if they have to modify the action of the cell, they have to have their receptors on the surface of the cell. These ligands have to work on the surface of the cell. Again, all those neurotransmitters or drugs or hormones which are large molecular weight and which are highly polar, or either they cannot dissolve into lipid membranes of the cell, they cannot enter into cell. Because they cannot enter into cell, so their receptors should be present on the surface of the cell. Their receptors should be expressed on the surface of the cell. So this receptor is cell surface receptor, or it is a protein which is expressed on the surface of the cell membrane. Opposite to that, there are some, you can say, ligands which can directly enter into a cell. They can directly enter into a cell. These are either small molecular weight substances, or they are highly lipid, yes, they are highly lipid soluble. Now such substances which are either very small or they are highly lipid soluble, right, they can dissolve into lipid membrane and enter into cell. And because they can enter into cell, so their receptors should be present, yes, within the cell. Their receptors should be present within the cell. That's the beauty of it, that all those substances which usually cannot enter into cell, their receptors are expressed on the cell surface. And all those substances which can enter into cell, their receptors are present within the cell. They may be in the cytosol or within the nucleus. Is that right? Now, next time if I ask you that there is a substance which is very large and highly polar, its receptor should be present on the surface. And if I say that there is a substance which is highly lipid soluble and which is very small molecular weight, its receptor should be present within the cell. The receptors should be present within the cell. So it means primarily we can classify receptors expressed on the surface of the cell and receptors which are present within the cell, right? So this is the basic classification of receptors. Now, I can give you some very interesting examples of those substances for which receptors are on the surface. For example, peptide hormones. For example, yes, you know many peptide hormones like, yes, insulin. Everyone knows about insulin. Growth hormone, prolactin, FSH, yes, LH, TSH, and so many others. All these substances are large peptide. They cannot enter into cell. So their receptors should be present on the surface of the cell. Opposite to that, another group, along with that, there is another group of compounds which are highly charged. For example, catecholamines. Catecholamines, for example, yes, epinephrine, right, or norepinephrine, or dopamine, right? All of these are highly polar. If these are highly polar, highly charged, can they enter into cell? No. So their receptors should be present on the surface of the cell. In the same way, either you can talk about acetylcholine, yes. There are 5-hydroxytryptamine or serotonin. There is histamine. There is prostaglandins, and there are so many substances. All of these are not soluble enough to enter into cell. So the receptors should be present on the surface of the cell. Opposite to that, there are some substances which are really highly lipid soluble. There are other substances which are highly lipid soluble. This should not remind you of facial nerve palsy, right? And small molecular weight substances or substances which are highly soluble, their receptors are present within the cell. What are the examples of such substances? Fine. All the hormones, the substances which are derivative of cholesterol. More truly speaking, all the substances which are called steroid hormones. Now, steroid hormones, yes, steroid hormones are highly soluble. Now, it means steroid hormones will work on the surface of the cell or within the cell? Answer is within the cell, right? Now, can you name few steroid hormones? Of course, there are so many. For example, yes, testosterone, progesterone, estrogen, yes. Then there is testosterone, progesterone, estrogen, then there is aldosterone, then there is glucocorticoids, and even vitamin D is basically a derivative of steroid-like structure, so we can say vitamin D. What is the special name for vitamin D? Cholecalciferol. What is the special name for vitamin D? Cholecalciferol. Is that right? Now all these substances are highly lipid soluble, so when they act on the target cell, they freely move through the membrane, and the receptors are present within the cell, and there they act, right? Now, then there are other substances which are very small molecular weight. For example, T3, T4. These are thyroid hormones. Or you can talk about retinoids. Retinoids are vitamin A related compounds. These substances are also able to cross the cell membrane, so their receptors should also be present within the cell. Now we will go into detail of receptors. We have already discussed that there must be two types of receptors. Number one, the receptors which are expressed on the surface of the cell, and receptors which are expressed within the cell. Is that right? There are receptors which are expressed on the surface of cell. They are for those molecules which are highly polar and large molecular weight. And then there are receptors which are within the cell, which are for small molecular weight ligands, or which are meant for highly lipid soluble ligands. Now, we will start discussing, receptors, we have said it, you can say within the cell membrane, and these are inside the cell. Inside the cells. Now, first I will discuss all the receptors which are expressed on the cell membrane, right? Now we will go into detail of those receptors which are expressed on the, surface of cell membrane. Now let's suppose this is a cell, right? And this is the surface of the cell. There are many types of receptors which are expressed on the surface of cell. I will just talk about the most important groups. One whole family of the receptor is like snakes. You see, they are made by a very large, you can say, peptide. and this peptide 7 times passes in and out of the cell membrane. Look, what is this? This is a receptor, and this receptor consists of a long peptide chain and which passes in and out through the cell membrane. How many times? 7 times, right? And it has one domain outside. This is extracellular domain, and it has one domain which is inside, and this is called intracellular domain, right? This group of receptors are called, because they look like snakes, right? They are called, very special name for such receptors, they call them serpentine receptor. Serpentine receptors. Serpentine means something like, yes please, snake. Something like snake, right? This is a serpentine receptor, right? And they have also one simple name for this type of receptor. These receptors are also called, yes, serpentine receptors, or how many times they pass through the membrane? 7 times. So we call it 7 pass receptors. So I really love this name because it's easy to remember that it 7 times passes. But my kids like this one. They don't know how dangerous is serpentine. So they think serpentine receptors are a good term. Anyway, it's a very big family of receptors, right? And they pass through the cell membrane 7 times, right? They have extracellular domain and they have intracellular domain. Extracellular domain is ligand binding domain. To this point, what will bind? Ligand will bind. Ligand will bind to this domain, right? And when ligand binds here, it activates this receptor. Is that right? When ligand binds on this, it activates this receptor. So this domain is called, what is the name of it? Ligand binding domain. Opposite to that, once the ligand bind on it and stimulate it, then intracellular part will start giving signals to the cell. It will start giving signal to the cell. So this part is called, yes, what should be the name of it? Because it is going to produce effects in the cell. So this part of the receptor, which is going to produce ultimate effect on the cell, this part of the receptor is called effector, yes, domain. So what did we learn? That 7-pass receptor, like other receptors, have ligand binding domain and they are having effector domain. Ligand binding domain is outside the cell, effector domain is inside the cell. When ligand bind with the ligand binding domain, there is a change in the receptor which is transmitted to the effector domain, and effector domain give very unique signal to the cell, right? Now, life is not so complex that everything has to pass 7 times. There are other group of receptor, whole family of receptors, where receptor is very innocent. It passes only once. Receptor passes only once through the membrane. There is no way to pass it 7 times. They passes only once through the cell membrane. Because they pass only once through the membrane, so their name is also very simple. They should be called, yes, 1-pass receptor. They should be called 1-pass receptor, right? These receptors should be called 1-pass receptor, right? Another way, of course, then what happens, that here is the ligand which binds here, yes, this is the ligand. Here is the ligand which bind with the ligand binding domain. When ligand bind with the ligand binding domain, then effector domain start giving, yes, signal to the intracellular environment. Is that right? What is this domain? Effector domain, and effector domain will start giving signals within the cell. These are 1-pass receptors, right? There are many substances which work on the 1-pass receptors, and there are many substances which work on the 7-pass receptors. For example, the substances which work on 7-pass receptor, there are so many. I will just give you a few examples like catecholamines. Catecholamines, every medical student knows there is epinephrine, there is norepinephrine, yes, there is dopamine. All of them work through 7-pass receptor. All of them work through serpentine receptors. Even do you know that acetylcholine has muscarinic receptors which are 7-pass receptors, which are to the family of 7-pass receptors. Even prostaglandins, they have 7-pass receptors. Again histamine, 7-pass receptors. You can talk about 5-hydroxytryptamine, that is also, which is also called serotonin, that also work on the 7-pass receptors. And then you can talk about, yes, there are so many substances like catecholamine, acetylcholine, prostaglandins, histamine, 5-hydroxytryptamine. Would you like to know that even TSH hormone work on the 7-pass receptors? So there are many substances which work through the unique family of receptors expressed on the surface of the cell, and this family is called serpentine receptors, or other name is 7-pass receptor. But one very interesting point which I would like to highlight here, all of these receptors are coupled with very unique type of protein within the cell. This protein is having alpha component, beta component and gamma component. This is a trimeric protein. This is a trimeric protein. It has 3 peptides. This trimeric protein, especially its alpha component, it binds with a special energy powered guanine GTP. You know, GTP binds with where? GTP is binding with the G protein. G protein consists of alpha unit, beta unit and gamma unit. So this black is the G protein and this is GTP molecule. If GTP molecule is binding here, it is very rich in energy and it becomes active. Or if one phosphate is broken, then it is GDP. Then again its energy level of GDP is low. So GDP attached, G protein is inactive. Now listen. Alpha, beta, gamma unit make a trimeric protein which can bind with GDP or GTP, right? If it is binding with the GDP, it is low energy level. If it is binding with GTP, it is a high energy level. And because these proteins are binding with the GDP or GTP, they are simply called G proteins. They are simply called, yes please, G proteins. Thank God some doctor did not put the name of this protein on his personal name or daughter's name. They simply call it G proteins. So what are G proteins? G proteins are unique type of intracellular trimeric proteins to which GDP or GTP binds. Is that right? And these are called G protein. Now remember one thing very interesting, that all the serpentine receptors are working through different type of G proteins. All the serpentine receptors work through G proteins. That is why there is a third name for these proteins, these receptor, and that is G protein coupled receptors. G protein coupled receptors. Now listen carefully. In your MCQ, if you see there is a word of serpentine receptor, we are talking about this receptor. If they mention 7-pass receptor, again it is the same system. Or they call it G protein receptors, it is the same system. Why they call it serpentine? Because it looks like a snake. Why call it 7-pass? Because 7 times its peptide passes through the cell membrane. Why they call it G protein coupled receptors? Because these receptors are intracellularly connected with the and working with the G proteins. Is that right? Now I would like again come back to 1-pass receptor. 1-pass receptors have a unique effector domain, and usually this effector domain is enzyme. Because this is enzyme, so other name for 1-pass receptor is enzyme linked receptors. Enzyme linked receptors. Now look here, it is 7-pass receptor, it is 1-pass receptor, it is G protein coupled receptor, it is enzyme coupled receptor. Is that right? Now we'll go to another last major group of the receptors which are present on the cell surface. That receptor is, okay, look here, yeah, what is here? This is a unique type of channel, right? And this channel is passing through, it is made of multiple peptides. Now what really happens? How this channel is made? This channel is made by the multiple peptide chains put together. For example, this is one peptide chain, here is another peptide chain, here is another peptide chain. In the same way, there is one peptide chain which is put over here. When you assemble them, all of them, right, they become a channel. Is that right? Now this is a channel which may allow certain substances to enter into cell or leave the cell. Usually these channels allow the movement of the ions, so we call them ion channels. Now remember, there are many types of ion channels, right? Some ion channels are controlled by the voltage, some are controlled by the voltage, so we call them voltage sensitive channels. We call them voltage sensitive channels. Then there are some other channels which are activated by the ligands, special type of ligands, and those channels are called ligand operated channels. So actually receptors are basically ligand operated channels. What really happens that in between the peptides there are unique type of pockets, and to these pockets a particular ligand may stimulate this pocket or another ligand may stimulate this pocket. Now look here, if these ligands are not there, channel is closed, but as soon as these ligands stimulate the channel become open, and depending upon the type of channel, certain anions or cations start moving across. Is that right? For example, now okay, we must know what is this substance. In this example we can say that maybe this is acetylcholine. You know, acetylcholine is a unique type of receptors which are called nicotinic receptors. Nicotinic receptors are basically ion channels, and they have 2 points where acetylcholine can bind. When acetylcholine binds over here, then what really happens, nicotinic channels will open and cations will start moving across and electrical properties of the cell will be altered, right? So this was a brief opening session about the receptors, group of receptors, families of receptors which are present on the cell surface. There are many others. We will not try to learn everything. We will only concentrate what is relevant with our exams, right? For exam viewpoint, it's very important to know about 7-pass receptor which are coupled with G protein. It's very important to know about 1-pass receptors which are usually linked with enzymes, and it is extremely important to talk about ion channels, ligand operated ion channels, right? Because they also act as receptor, because a certain ligand will bind here, they will undergo some change and biological action will be produced. So we were talking about receptors, and we have already classified receptors as receptors on the surface of cell, that is they are present on the cell membrane, and receptors which are, yes, intracellular receptors which are within the cell. Then we have discussed the many classes of the receptors which are surface of the cell. Out of that we have seen in our previous diagram 3 types of receptors. One group was, yes, serpentine receptors, or other name for that was 7-pass receptors, or another name for that was G protein, yes, coupled receptors, 7-pass receptors or serpentine receptors. So this was one group of receptors which are expressed on the surface of cell. Second group of receptors which are expressed on the surface of the cell was, yes, and 1-pass receptors, or this was also called enzyme, yes please, enzyme linked receptors, right? Then we have talked about third type of surface receptors, and third type of receptors which are transmembrane, they are called ion channels, but not every ion channel, only those ion channels which are ligand operated, ion channels, because there are many other channels in our cells which may not be operated by ligands. They may be leaky channels which may be leaking all the time, or they may be voltage operated channels, right? Out of these channels, only ligand operated channels should be considered receptors for specific ligands. Now look, my very dear students, we are going into detail of serpentine receptors, right? And this is one area, if you really understand very well, right, this is going to make your life easy about physiology, about biochemistry, about pharmacology, and even some aspects of pathology, right? So let's go into detail of serpentine receptor, I will make a diagram and we'll be just working on this concept that what are serpentine receptors, how do they work with G proteins, and how G proteins lead to the intracellular function. Right? Everyone is ready to understand it with open mind and closed mouth? Okay, but keep your eyes open at least. So let's suppose here is a cell and this cell is, okay, this is very big cell, but now this cell must be having some serpentine receptors. Let's suppose that there is a unique type of serpentine receptor here, and this is its intracellular domain. Right? Now what really happens? Let's suppose this receptor is coupled with a unique type of G protein. First of all, ligand will come and bind here. Suppose this ligand is epinephrine. We are just taking an example. This is epinephrine, and this serpentine receptor is present on myocardial cells. So this should be, yes, beta 1 adrenergic receptor. So this serpentine receptor is beta 1 adrenergic receptors. Now you must be knowing that beta 1 adrenergic receptors, which are present on the myocardial cells, they are intracellularly coupled with G stimulatory proteins. Let's see what is G stimulatory protein. Look, what are G stimulatory proteins? As I told you previously, G proteins are made of how many peptides? 3 peptides: alpha, beta and gamma. There are 3 peptides, right? And they do have, alpha unit has one unique pocket to which when it is inactive, it is holding, what is this? GDP. I mean, when GDP is binding with the alpha subunit, it is not functional. And when alpha G unit is not functional, it expresses special domain, and through these domains, it loves to keep a good relation with beta unit, and with the, yes, gamma unit. So until GDP is there, alpha protein is at low energy level and it is making a friendship with beta and gamma unit. You know, when you have less money, you have more friends, isn't it? You are moving with the friends and you are very happy with them, isn't it? So now alpha is poor. So it is having very good relation, a very humble relation, very loving relation, very caring relation, very attachment relation with beta and gamma unit, and these 3 friends are together with little energy at GDP level. Now, but alpha is clever. All the time it has special antennas, special domains, which are all the time looking for, is there any special message through this receptor? Look, whenever this serpentine receptor is stimulated by epinephrine, this part of receptor, look, this was effector domain. If effector domain is folded like this, then alpha cannot interact with effector domain. But as soon as, what comes here? Ligand. As soon as ligand comes here, this receptor changes its effector domain into this fashion. It means the effector domain from inactive form has gone to active form. Again listen. When ligand is not there, effector domain is inactive. As soon as ligand is there, effector domain is active. When effector domain is active, it means it has received a message from active domain, can stimulate what? Alpha subunit. So alpha unit becomes stimulated. It gets a very special message from abroad, from outside, from overseas. It sees a very special signal. When effector domain is stimulating alpha subunit, first of all, it will lose, yes, what will it lose? It will lose GDP, and it will acquire very high energy molecule. What is it? GTP. It means that as soon as, look now, as soon as ligand bind with the receptor, ligand binding domain is loaded, receptor undergoes a change, this stimulate effector domain stimulates G protein alpha subunit throws away the low energy molecule and now accommodate high energy molecule. Now it becomes rich, and when it is rich, it behaves the way some of you behave, that it is pulled away its hand from its long friends. It's no more interested. It has taken its hand to the pocket, no willing to have a relation. And beta gamma become very sad, and alpha thinks it is fully energized, and it detaches from there. Alpha will detach from there. So friends will go other way. They are very sad. Which friends? Beta and gamma. They are with each other, weeping all the time that the rich alpha has left them energy rich, and alpha is very, very happy because it has high energy. Now this alpha subunit very, very happy with high energy, having what substance here? G, D, GT, GT, P. When it has GTP, it is a high energy molecule, and it is no more interested to have a relation with beta and gamma. And it wants to develop new relations. With whom? There's a clever man here who is waiting for it. Right? Look. There's another protein which is hanging to the interior of the cell membrane. And this protein is called, adenylyl cyclase. And this is very, very clever protein. You see? It is having a unique point here. And as soon as alpha unit is stimulated, it will love to have relation with which? Adenylyl cyclase. It will go and fit into adenylyl cyclase and stimulate adenylyl cyclase. As soon as adenylyl cyclase is stimulated by a stimulated GTP loaded alpha component, adenylyl cyclase will give a very unique signal to its a special domain, and this protein will become active. This enzyme will become active. Look again. As soon as alpha unit stimulate adenylyl cyclase effector unit become active, and when it becomes active, it breaks down the ATP into, cyclic AMP. It means in the cell, cyclic AMP level start going up, right? Now, but there's one sad thing. What is that? The sad thing is that, you see, it has been bad to its friend. It has kicked its friend and tried to make a high level friendship. Now, what it will do? It will have a hammer here. And it will activate a unique point within this protein. This unique point in this protein, when it is activated by this hammer, by a domain of adenylyl cyclase, you know what it does? It breaks down GTP. Again, listen, its point is very important to understand. When it will go and stimulate adenylyl cyclase, as soon as it has stimulated adenylyl cyclase, let me revise it another way, that alpha unit, when it was stimulated, right, it required GTP and stimulated adenylyl cyclase. Adenylyl cyclase converted ATP into cyclic AMP. Fine. But meanwhile, what really happens? As soon as alpha unit has stimulated adenylyl cyclase, alpha unit has a unique type of intrinsic domain. This intrinsic domain will break down what? GTP. And now what happens? That one phosphate goes out. So guanine triphosphate has lost one phosphate. It is no more triphosphate. Now it is guanine diphosphate. So one phosphate will come out. So it means while it was stimulating adenylyl cyclase, its intrinsic GTPase is activated, which is the internal enzyme of this molecule, and break down its GTP into GDP and phosphate. And as soon as this GTP become GDP, now it is back to its lower energy level. And when it is back to its low energy level, now it is very, very, sad. It is very, very sad. And when it is sad, it is no more interested to stimulate that thing, right? It will detach. You know where it will go? It will go in search of its old friends, right? So what really happens? That it will come back, and old friends are old friends. They are always good, and they are waiting for their friend, so it will come back and it will again attach, it will make again good friendship with them, and it become again happy, but its energy level is high or low? GDP, it's that energy level low. As soon as these friends become happy, three proteins become happy, they again start moving, and nature can't be changed. Again it will find that if there is some message from outside, some ligand is binding there, a serpentine receptor is activated, and again it will get stimulated. It will again lose the GDP, acquire the GDP, become very energy rich, kick the beta and gamma away, become very active in search of a new effector system, stimulate that, lose its GDP into GTP, go back. Meanwhile, this stimulated protein lead to the conversion of ATP into cyclic AMP. Is that right? Now, originally what happened? Doctors or scientists only knew one thing, that when they put, what was this? Epinephrine. Doctors knew whenever they put epinephrine on a cell, in the cell, cyclic AMP level goes up. 30 years back, 40 years back, only doctors knew one thing. They put the epinephrine on a cell, and in the cell what levels go up? Yes, cyclic AMP levels will go up. Is that right? Cyclic AMP levels will go up. Then doctor thought that this is the first message to the cell. This is the first message to the cell. And they thought this is the second messenger in the cell. So actually, from those days, the term for ligand was first messenger. First messenger to the cell, that its action should be modified. And they said once the first messenger act, they never knew these things. They just knew when first messenger act on the cell, in the cell, level of what will go up? Yes, second messenger. But now, thank God, we have solved a lot of mystery in between. We don't say there is first messenger and then there is high level of second messenger. We know what really goes in between. Actually, first messenger stimulates serpentine receptor. First messenger bind with ligand binding domain. Ligand binding domain will lead to stimulation of intracellular effector domain. Intracellular effector domain will stimulate alpha component of G protein. And this G protein alpha component will go and stimulate adenylyl cyclase. Now listen carefully. This G protein, whose alpha component can stimulate adenylyl cyclase, we call it alpha stimulatory. This beta and gamma stimulatory. So whole of this is called which G protein? Whole of it is called G stimulatory proteins. So in a simple way, we can say ligand bind with serpentine receptor, and serpentine receptor is coupled intracellularly with G stimulatory protein, and once G stimulatory protein is stimulated, it loses the GTP, acquire the GTP, and alpha unit with GTP goes and stimulate adenylyl cyclase. The other name for adenylyl cyclase is, effector molecule, effector protein. So what we can say, the first messenger stimulates the receptor stimulates the G protein stimulate effector molecule increase the level of second messenger in the cell. Is that clear? Now, what the second messenger will do? Second messenger will stimulate a very unique type of protein, and that protein is, intracellular cyclic AMP is a form of special signal. This will stimulate a unique type of enzyme. Cyclic AMP will stimulate a unique type of enzyme, and this enzyme is called, I will not tell you the name of this enzyme. You have to tell me the name of this enzyme. Very, very clever enzyme, right? Now, cyclic AMP will stimulate this enzyme. When this enzyme is stimulated, it becomes very active. And you know what it does? This enzyme will start, it will take some intracellular protein, it will attack, this will attack on some intracellular proteins. This enzyme will attack certain intracellular proteins, and when this enzyme will attack the intracellular proteins, what happens? That it will add phosphate to them. Look here. What it is doing? Actually, let's suppose there is an enzyme here. There is an enzyme here, right? This protein will act on that enzyme and enzyme become, wow, it become active. Here it was inactive. Why? Because it has added phosphate to that. It means this protein, this green protein, has phosphorylated this enzyme. And when enzymes are phosphorylated, their action alter. When their action alter, it means metabolism of the cell alters. Metabolism altered. Right? Again. Rising cyclic AMP stimulate the green protein, and green protein stimulate enzymes. How? Rather, it modifies the action of enzyme by phosphorylating the enzyme. When enzyme gets phosphorylated, what happens? That their action alter. When enzymes action alter, cell metabolism alters. This is one way. Second is that some of these can also act on another group of proteins, and the second group of proteins are fine. These are certain types of transcriptional factor which can regulate the genes, and this enzyme will lead to phosphorylation of transcriptional factor. When transcriptional factors are phosphorylated, they can work on the nucleus. They can work on the nucleus, and that will result in altered genomic expression of the cell. Genetic machinery of the cell is altered. Again, this will stimulate some transcriptional factor or special unique protein which can translocate to the nucleus. You know, in the nucleus there is genetic program of a cell, and so cell's genetic program is altered and genomic expression is altered. So what is the result? Altered genomic expression of cell. Genomic expression of cell. Cell may make new proteins, right? Then not only this, even one more very important thing is that this green enzyme can activate, you see now, what is this? Here is a ion, yes, channel. Now what really happens? This can lead to the, yes, phosphorylation of ion channel. And if ion channel is phosphorylated, what really happens? Its function also alters. And if ion channel's function alter, it means there will be change in electrical properties of the cell. For example, if this is a calcium channel, and if this calcium channel is phosphorylated, it will open up. And you know there is more calcium outside the cell and there is less calcium inside the cell. So naturally, when calcium channels are phosphorylated and they become more functional, then extracellular calcium start moving in, and cell is being loaded with cations. When cell is receiving the cations, so it become electrically more electropositive and electrically stimulated. So what do we really see? We see that ligand is the first messenger, bind with the ligand binding receptor domain, that stimulate intracellular effector domain. Effector domain stimulate G protein, especially which component of G protein? Alpha component. It loses the GDP and acquires GTP. GTP driven, right? Reject the beta and gamma units and rush to the adenylyl cyclase, stimulate the adenylyl cyclase. Meanwhile, it is itself inhibited by breaking down its GTP into GDP, and it has to recycle back to its friend and again play the role again. Meanwhile, adenylyl cyclase will convert ATP into cyclic AMP. Cyclic AMP will stimulate a special green protein. You have to tell me the name of this green protein, isn't it? So this green protein can phosphorylate enzymes and alter the cell metabolism, phosphorylate the transcriptional factor, alter the cell genomic expression, phosphorylate the channels and alter the cell's electrical properties, right? What is the name of this green? Okay, what is the name of this green protein? Yes, you have to tell me. You have 10 seconds to tell me what is the name of this green protein. Yes. What is the name of this green protein? Who is going to tell me? Yes. What is this? This is supposed to phosphorylate target proteins. And because this can phosphorylate target protein, so such proteins are called protein kinases. These green proteins are called protein kinases, and because they are dependent on cyclic AMP, so they are called protein kinase A. They are called protein kinase A. So you must know what is protein kinase A. Protein kinase A are unique type of enzymes which are present in the cell, and when these enzymes are stimulated by the high level of cyclic AMP, they will lead to phosphorylation of target protein. Some proteins are enzymes, alter the cell metabolism. Some proteins are transcriptional factor, alter the cell genomic program. Some proteins are ionic channels and alter the cell's electrical properties. Is that right? So in a short, first messenger, receptor. These G proteins are also called biological switchers. They are called biological switchers. They switch on or switch off the biological system. So first messenger, receptor, biological switchers, effector protein. What is that? Effector protein. Then second messenger, and after that protein kinases and their action on the target cell, altering the many activities in the cell. Now you see, there was one hormone or neurotransmitter came here. There was one molecule. It could activate many it could activate one receptor for a long time. This may activate many G proteins. Many G proteins may activate many adenylyl cyclase. They convert thousands of the ATP into cyclic AMP. They activate thousands of the protein kinases. And protein kinases phosphorylate millions of the proteins. It means just one ligand could bring changes in millions of the molecules. You can say this is a biological amplifier system. What do we call it? Biological amplifier system, that when first messenger come, second messenger are beyond. Biological effect is so much amplified that very small levels of neurotransmitters and very small levels of the hormones are enough to produce great changes in the target tissues and cells. To understand one more thing, that as this is a special biological amplifier system, right, there are also counter-regulatory mechanisms. For example, what we say, that this particular ligand came, an intracellular cyclic AMP was increased and certain effects were produced. Now these effects can be countered in many ways. One of the way to cancel these effects or counter these effects, right, or to nullify these effects is another G protein system and their associated receptors. Let's suppose there is another. Now this is a different type of serpentine receptor, right? It is also serpentine receptor, but you see its ligand binding domain is, this domain is, look here, it is different than this domain. So this is a different serpentine receptor and that is a different serpentine receptor, but both of them belong to the same serpentine receptor family, right? So what really happens, that in this particular, here the ligand was epinephrine. Maybe on that side ligand is not epinephrine, ligand is, what is the ligand here? It may be acetylcholine. So acetylcholine can stimulate this receptor, and this receptor, serpentine receptor, it is binding with a different ligand and it will give signal to intracellular G protein of a different type. There in previous example it was G stimulatory. Here there is G inhibitory with alpha inhibitory, yes, beta inhibitory and gamma inhibitory. Now again the story is same, that when normally it is having here GDP, but as soon as ligand bind with the receptor, receptor stimulate the G inhibitory, then G inhibitory loses GDP and it acquires, yes, what? GTP. So it becomes active, and when it becomes active, right, it also has its effector component and its effector component is going to work on a, yes, it is going to work on different domain of adenylyl cyclase. This is entirely a different domain, right? And it will stimulate that domain, and when this domain is stimulated, what really happens that this particular domain stimulation will lead to inhibitory action on the effector part of adenylyl cyclase. So what we can say that when alpha stimulatory come, adenylyl cyclase is, yes, stimulated, and when alpha inhibitory come, then adenylyl cyclase is, yes please, inhibited. So naturally we can say this is one of the central molecule, this is the effector protein. When G stimulatory comes, adenylyl cyclase is stimulated. When G inhibitory comes, adenylyl cyclase is inhibited. It means that G stimulatory by stimulating adenylyl cyclase increases cyclic AMP level, and G inhibitory or alpha inhibitory by inhibiting adenylyl cyclase inhibit the level of cyclic AMP. It means G stimulatory system activates protein kinase A system, and G inhibitory system inhibits protein kinase A system. So what we can say that ligand number 1 epinephrine and ligand number 2 acetylcholine, they are having opposite effects. Even their receptors are also having opposite effects. This is cholinergic receptor of muscarinic type, and this adrenergic receptor is coupled with G stimulatory. Here muscarinic receptor acetylcholine is coupled with G inhibitory, and this G stimulatory system increases cyclic AMP and G inhibitory system decreases cyclic AMP level. So what really happens that cyclic AMP level will drive the activity of protein kinase A, and that will determine the phosphorylation of certain enzymes and protein kinases and target cationic channels. So that will eventually result in altered metabolism or altered genomic expression or altered electrical properties. Is that right? One thing which is very important is that here when alpha inhibitory detaches and work with the adenylyl cyclase, these two friends are very angry. These two friends are very angry, and they also get involved in some negative activity. That was beta inhibitory and gamma inhibitory. You know what they do? They bind with a very special type of, you can say, potassium channel. What is it? This is potassium channel, and this potassium channel has these two pockets on the side, right? And when these angry beta gamma units, bind with these potassium channels become active. And when potassium channels become active, you must be knowing cell is very rich in potassium. So potassium start moving through the channel and it start leaking outward. So it means that beta gamma unit, inhibitory unit, will lead to potassium efflux, loss of potassium out of the cell. And if cell is losing the potassium, it means it is losing the cations. If it is losing the cations, it means it is becoming more electronegative. It means cell is becoming more hyperpolarized. It means cell is electrically inhibited. Now compare this with that. Here cationic channels were stimulated, there also cationic channels are stimulated. But what is the real difference? Here cation is calcium, which is more extracellularly. So when this channel is phosphorylated, calcium moves in. So positive ions come in. When positive charges come in, cell gets stimulated. But in that case, once the channel open, potassium is more inside, less outside. So from high concentration, from inside they move outside. So cell is losing the cations, so it becomes electrically negative. So what we can say now, if you look at the whole picture, what you really see? You see that epinephrine opposes the action of acetylcholine. They oppose the action of each other. This serpentine receptor opposes the action of that serpentine receptor. G stimulatory opposes the action of G inhibitory. One is increasing the cyclic AMP, other is decreasing the cyclic AMP. One is stimulating protein kinases, other is inhibiting protein kinases, right? So it means both of them are having counter-regulatory action. Is that right? Now